in the US and 9 o'clock Karachi time. Assalamu alaikum, everyone. And today it is, I think, the seed for Nobel Prize for Pakistan is being sown by the galaxy of research stars and the rainbow of scholarly activity. It's kind of very heartening to know that a group of scientists and great minds in Pakistan have come together to give us this presentation of their work. And Dr. Izhar Hussain is the Executive Director of Institute of Biological and Biochemical and Pharmaceutical Sciences. And so that encompasses the entire spectrum of what we are, where Pakistan is starting to go. And this is the really heartening for us to see the young people coming forward in the science. And Dr. Izhar will introduce the team. What their roles is, I'm just going to name them. Dr. Talat Roome is the associate professor and the director of the Institute of Advanced Biological and Animal Research, and Dr. Maria Rizvi is advisor of Dow Institute of Life Sciences, and Dr. Mushtaq Hussain is a professor and principal of the Dow College of Biotechnology, a very important position, and Dr. Sadia Asim, Miss Sadia Asim is the Director of Institute of Biological, Biochemical and Pharmaceutical Sciences, working with Dr. Izhar. So without further taking up the precious scientific time, I'm going to ask Dr. Azhar to please introduce his team, and we are very anxious to hear about their achievement and their thoughts and their advances. Over to Dr. Azhar Sahab.
Thank you, sir. Thank you very much. First of all, I want to thank Merit APPNA for providing this opportunity, and also thankful to you for moderating this webinar. And if I will not say that how Dr. Uzair is encouraging us to interact with APPNA and in other forums as well. So I think it will be something which I could not do. So thank you very much, Dr. Uzair. So we will start this webinar by Dr. Talat Roome. She will introduce her pre-clinical and animal studies she's conducting. Dr. Roome is a PhD, and he is also a scholar. She did her postdoc in USA, and then she will introduce her work. She's making models and doing work for the industry as well. And nowadays she is working on anti-rabies vaccine production in Pakistan from BASIC. So if that vaccine production unit do its work successfully, it will open a door for other vaccine productions as well. She will be followed by Dr. Sadia Asim. She will introduce about her work about clinical trial and bioequivalence and bioanalytical things. We have started working under her in clinical trials. We conducted 2 clinical trials. One was FDA approved clinical trial. The second was Chinese clinical trial, and third one, which is coming, is also from FDA approved trial. And we cannot work on bioequivalence and bioavailability studies because of some legal lacunas in Pakistan, on which Dr. Uzair and Dr. Zulfikar Bhutta is trying to remove that lacuna, in which they require COPP and GMP certificate from the patent holder. So that is a problem which, because of which we are losing many. We have about 26 applications for clinical bioequivalence studies, but we cannot conduct because that particular thing. She will be followed by Dr. Maria Rizvi. Dr. Maria Rizvi will talk about the vaccine and sera production unit in DAW, and that is a unit that is second facility for biological production in Pakistan, licensed by Drug Regulatory Authority. we are looking for more products to make it more commercially viable. So we are ready for toll manufacturing as well. And then the Professor Mushtaq, who is a really, we hope, going to be a candidate for Nobel Prize in future. And he is working on many things, particularly mRNA vaccine, which is a new area in vaccine production. And I think that will be an area which is very important for us and for the whole humanity. And I would like also to introduce that in Dow University of Health Sciences, we have all clinical surgical units available. We have cyber knife facility. We have also the uh, liver, and bone marrow and kidney transplant unit, and all. We have about 16 schools and institutes and colleges relating to healthcare, like bioequivalence, pharmacy, medical technology, and School of Health and School of Nursing. And we also have a school Institute of Behavioral Sciences, which is also very important because they have the very big team of those who are working on behavioral sciences and psychiatrists and psychologists. It is one of the biggest unit which have psychologists and psychiatrists, which is a rare species in Pakistan, and I think all over the world. So this is the thing. We have about 1,000 beds available, about 200 beds in ICU. We have a specific infectious disease hospital, and that is considered as one of the very uh, sought after hospital during COVID. Even those who had lost their loved one due to COVID, they were praising the quality of work they are doing. And then we also have large OPDs. About 3,000 patients are coming daily to our OPDs. So it is a very big area which we can contribute in clinical trials. So it is a government entity. We are not commercially and we are not profit oriented. So that is another thing which I would like to discuss, but not going. I think the many things will come out by the presentation which will be made. So I request Dr. Talat Roome to start her presentation. Uh, Thank you very much, Dr. Sarwat Hussain Saab. Dr. Talat Roome, please.
Dr. Saab. Yes, I'm starting. Assalamu alaikum, and thank you very much, Dr. Izhar, for the generous word, and I'm thankful to Dr. Sarwat and Dr. Izhar for giving us this opportunity. Today, I'm representing Daw Institute for Advanced Biological and Animal Research, which is a hub of animal-based research carrying vast array of essays related to preclinical and toxicological studies. This institute is actually a well-equipped facility that deals with the broad aspects of biological, biomedical, and pharmaceutical basic research and applied sciences. We actually focus on mechanistic evaluation of disease pathogenesis and therapeutic entities at the level of biochemical, cellular, and molecular. Preclinical and disease model can be designed on genetically modified transgenic and wild-type rodents, non-rodents, and non-human primate. Toxicological studies protocol have been well established in the institute as per FDA-approved guidelines. And at the last, we focus on development of biologicals, specifically upstream processing like anti-sera, anti-snake venom, hyperimmune plasma we are producing. And as Dr. Izhar already mentioned that we are working on a vaccine, and our current target is a rabies, anti-rabies vaccine, and leading it to the mass scale production. Our panelist and colleague, Ms. Maria Rizvi, will share about mass scale production. We believe to provide a lab facility to assist researchers in their efforts of quality research with respect to use of laboratory animal and act as a resource center for researchers focusing on the principle of animal care with compliance of the guideline and SOPs. On the basis of our progressive development and strategic plan, this institute comprises functionally distinguished sections as a interactive entities. And this institute actually governed by the different well-trained, specialized and skillful professionals. In Central Animal House facility, we have 3 veterinary doctors having DVM degree and 2 of them having PhD in microbiology, one having a microbiology degree and other DVM veterinary doctor has a specialty in animal nutrition and feed. As we mentioned, we are producing anti-venom sera in horses, so we have one equine specialist. Basic biological manufacturing facility, we have 2 important components: venom collection and processing laboratory, and plasma collection and processing laboratory as per WHO guideline. And we have a well-defined quality operations sections. We have a quality assurance executives, quality control managers, officers, production managers, and all are the PhDs. And we have a staff scientist to look into the basic science troubleshooting, and we have a scientific officer in this section as well. Today my talk main focus will be preclinical and toxicological contract labs. So in this section we have 3 PhDs and one is a molecular medicine PhD in a field of molecular medicine, one is a proteomics specialty, and one staff scientist has expertise in the area of material sciences, and 2 the scientific officers also incorporated in this section. DIABAR has established, implemented, and maintained the quality management system via incorporating the components of resources, process and document control system, management responsibility, design control, and most importantly, corrective and preventive actions. This institute is operating under 230 SOPs that is related to the animal husbandry, management, breeding, and biosafety, biosecurity, management of waste management, additionally the clinical and preclinical toxicological studies protocol, production protocol, quality control, and of course lots of SOPs related to quality assurance. We adopt the standard of animal research following the international guidelines and policies to perform the preclinical study, and we are establishing DAO animal care and use guidelines, and up till we have developed 20 DAO ACUGs. The central area of DIABAR comprises 4 acres that established first in 2008, and in continuation of innovation, various new sets of the laboratory has been established. In our animal house facility, we have different strains of animal mice, an MRI strain, biopsy, and we have star rats, guinea pigs, hamsters, and transgenics knockouts as well as per demand of the studies. We have horses, as mentioned, we are producing hyperimmune plasma against venom in horses. So we have horses, we are gradually increasing the strength as per the demand of the vials that we have to produce commercially. We deals with the 4 different snakes, big 4, cobra, crate, Russell viper, and soy scale vipers. We have rabbits and rhesus monkey for the challenge studies. Briefly, I would like to cover a few successful projects, preclinical model that we have established in our facility. We established hyperlipidemia and atherosclerosis model in apoe knockout mice. Various immune and inflammatory models has been established in the wild type animals. We have developed hepatocellular carcinoma through the DEN model, and we are currently extensively working on excisional and incisional bone model that I will share in next few slides. We are working on the arthritis and osteoarthritis. We have a adjuvant-induced arthritis model. We are working on the pharmacokinetics and dynamics and targeted delivery of nanomedicine. Recently, pulpitis and periapical murine model has been developed in this institute, which is very difficult, and we identified the novel target of these two pathogenesis in which we have introduced dectin-1 and osteopontin gene expressions, and we are working as a center for commercial testing. This is the lab facility we have in the institute. We have a molecular lab, in vivo pharmacology lab, animal surgical lab, animal cell culture lab, and various other facilities allied with this institute. That is a DRIPS, Daw Institute for Research for Biotechnology and Biological Sciences. Here we have a human cell culture facility, chromatography, flow cytometry, and for the molecular profiling laboratory and histopathology. We have a very good setup, and expert histopathologists are available to support the research projects and testing facility in DDRL, Diagnostic Reference and Research Laboratory. I would like to showcase few of my research. We purchased 6 pairs of APPNA knockout mice from Jackson Laboratory, USA, and the team in the institute developed 400 colonies of these mice, and we have conducted various research projects, and we developed hyperlipidemia and atherosclerosis, and here you can see that the blood vessel where hyperlipidemia and atherosclerosis has been clearly developed, and various test molecule and drug osteopensin and simvastatin nanoformulation we have tested, and its role has been evaluated against the atherosclerotic phenomena. We have developed adjuvant-induced arthritis model. You can see that how the arthritis model has been developed, and this is the X-ray, and we have a facility for the X-ray and MRI of the animals, and you can see that arthritis and the swelling and the destruction of the bone and damages and deformation of the bone, and in the presence of the test compound. Here I am not showing the new therapeutic entity, but I'm showing few standard drug, endomethacin and dexamethasone, how it is subsiding the arthritic effect. Not only we have tested MPP expressions, MPP9 expressions, and we have identified the level of reactive oxygen species in serum and plasma, and we have taken the synovial tissue and we tested the ratio of M1, M2 macrophages, and various genes expression has also been evaluated. Here is the data, and this has been recently published from this institute. We develop in vivo anti-cancer model utilizing DEM, and all 4 different stages of liver cancer development has been well established. We are working on bone model. As I mentioned, we are targeting splinted excision bone model, non-splinted models, chronic and acute, and a burn model. And at the moment, our prime focus to work with the nanofibers, nanofiber scaffolds, hydrogels, and this is the few example of the testing. Here is the data that we produced from this institute, and this paper also been recently published, and this is the histopathological evaluation of the bone, and we have also performed immunohistopathology, and we tested the marker of CD30, which is one of the bone healing marker, and here is the microscopic evaluations. We have a very good setup for Power Lab, and here the tested molecule can be evaluated against the spontaneous contraction of the tissues, and here you can see the antispasmodic activity of the tissues. So utilizing this setup, we can work on various receptors and ion channels, and we are collecting, processing, and characterizing venom, not only for the immunizing the horses, but venom is an attraction for us, because venom is a very important and attractive therapeutic modality and cosmetic as well. So we are working currently we are working antiviral activity of the venom. And we tested this venom against the avian influenza virus, and the team is further working on it. And CRO, if we look into the global CRO services market analysis, 20% market is as a preclinical CRO, which is gradually improving. And so we are improving our strength as well. And this is our toxicological studies. We are following OECD guideline for the skin sensitization test, a vast array of the biochemical and molecular profiling and histopathology testing and genotoxicity we are doing in this subject. And these are the multiple tests. I can add few of the tests, but we have established more than 30 tests as a CRO under the category of the preclinical and toxicological testing. As we mentioned, we are developing biosimilars, anti-sera, and vaccines. So up till different batches, having multiple immunization, we have produced thousands liters of hyperimmune plasma. And our manufacturing raw material is one of our landmark. And here you can see a few glimpses. The team is collecting venom from the snakes. And here you can see that the horse immunization, collection of the blood and the plasma, and the plasma showing titer against the venom, and IgG values has been calculated, and then it is tested for the potency assays and neutralization assays and all. As Dr. Izhar mentioned during introduction, that we have recently received anti-rabies vaccine development grant from Higher Education Commission, and we are at the moment at the phase 1. We are indigenously developing this vaccine, plasma-free vero cell line-based vaccine, and at the moment we are isolating virus and propagating in the mouse brain and neuroblastoma cell line. And our first target is to develop the virus bank. And our quality control team also doing good. And these are the few tests like bioburden, viral load, bacterial endotoxins, and neutralization in vivo and in vitro potency. They are performing as per standard guidelines. These are the few successful papers. Recently, these two papers published this year, few months ago. We have, as I mentioned, we have discovered the role of toll-like receptor activin and osteopontin in murine pulpitis model, and we have investigated photoactive curcumin zinc nanoconjugate and its potential in wound healing. The pharmaceutical herbal pharma industry, medics, their best-selling drug, digascolic drug, they sent to us for the testing, and 2 papers published. And we tested their product against genotoxicity and chronic toxicity, and we tested their product and polyherbal formulation for their antispasmodic activity. That paper also published. We published anti-cancer activity. We published a series of paper and mechanistic expression and the mechanism involved in the disease pathogenesis of arthritis and osteoarthritis. DRAP team, Dr. Abdul Rashid Saab visited us, and we have submitted our application for the preclinical licensing, and it's with the DRAP at the moment. And we have a collaboration and signed MOU with different pharmaceutical industries in Pakistan, and we are providing CRO services to them. And we have a lots of national and international collaboration, which I have listed here. This is my team. And thank you very much. This is a beautiful campus of the University of Health Sciences.
So next, Dr. Sadia, you have 15 minutes.
Okay, I will try my best to complete the presentation within time. Okay, this is Dr. Sadia Asim. I'm Director, IBBPS. I'm heading the Contract Research Organization and Biochemical Study Center at Dow. By profession, I'm a pharmacist and I'm associated with the clinical research field since 2007. Before going to my today's presentation, I would like to share a small IBBPS documentary that will give you a brief overview about the organization, about the scope of the work, and the facilities we have in the DAO umbrella. Here we go.
DAO IBBPS, Excellence in Bioequivalence Studies. The Institute of Biological, Biochemical and Pharmaceutical Sciences offers bioequivalence and bioavailability study services to local and international pharmaceuticals. These services are for all pharmaceutical products, both oral and subcutaneous dosage forms, with single or multiple API, that is, fixed dose combinations. We have a vision for improving the quality of healthcare services by providing clinical and pharmaceutical innovation. Let's look at the facilities at IBBPS. For volunteers in BE studies, IBBPS has a healthcare unit which is equipped with smoke detectors and security cameras. A team of consultants is available as principal and clinical investigators for these studies. A 24-hour ambulance service is also available in case of any emergencies during BA or BE studies. As per WHO Annex 9, the following facilities have been established at the IBBPS Bioequivalence Center. Access-controlled volunteer confinement area with separate wards for men and women. Recreational area equipped with indoor games. Drug administration area. Documentation area for randomization and blinding of studies, phlebotomy and urine collection area, blood sample processing facility. Access-controlled pharmacy, equipped with freezers and stability chamber. Emergency room, archive area, dining hall and kitchen. Importance of bioequivalence. Bioequivalence studies test two drug molecules that have the same active pharmaceutical ingredient and that release the substance in the same amount at the same rate and produce the same response. These studies help Pakistan's pharmaceutical industry export to and register their products in foreign markets that have stringent regulations. BE studies also assure doctors that the generic drug has the same efficacy and potency as the brand name drug. IBBPS provides these services to both local and international pharmaceutical companies. Bioequivalence at DAO IBBPS. Our rigorous quick turnaround bioequivalence studies help pharmaceutical companies bring their products to market faster. IBBPS is approved by the Drug Regulatory Authority of Pakistan and follows WHO, ICH and FDA regulation and ICH GCP, GMP and GLP standards and analyst proficiency testing by LGC UK for conducting BE and BA studies. IBBPS has the rare advantage of being situated within a world-class university hospital and medical research centre, Dow University of Health Sciences. With a proud heritage and a strong reputation, Dow University of Health Sciences is at the cutting edge of clinical and pharmaceutical research and innovation in the region. IBBPS has access to Dow University's 1,000-bed tertiary care hospital, along with its ISO 17025 and PNAC certified bioanalytical lab, ISO 15189 certified diagnostic lab, and intensive care unit. IBBPS provides the following services: protocol design and other studies related document design, medical and scientific BE report writing, statistical analysis, method development and validation of bioanalytical test methods, clinical study setup, bioavailability and pharmacokinetic studies, comparative dissolution profile, biowaiver preparation, in vitro and in vivo drug analysis, The Independent Institutional Review Board at DUHS ensures the rights and welfare of human subjects involved in research. The Institute has a pool of healthy volunteers for participation in studies and stringent protocols for taking care of volunteer recruitment, screening and selection, randomization of subjects, and assigning codes to investigational products. For recruitment of healthy subjects, the Institute follows WHO, EMA, ICH, and FDA's inclusion and exclusion criteria, including standard BMI limits. IBBPS is on a mission to provide the best solutions for cost-effective drug development and testing, so that quality pharmaceutical drugs are available to the public.
Thanks for watching a small documentary of IBBPS. Now I'm moving towards my today's presentation. Here I will tell you the scope of the work. Being CRO and the site management office, we are performing the following tasks, like including the clinical trial execution, medical writing, including the study protocol, informed consent form, case report form, investigation brochure, as well as the subject information sheet. And in addition to the medical writing, we are also reporting the pharmacovigilance to the DRAP and also to the IRB. We are performing the data collection analysis by using the EDC tool. For example, RedCap, we are using a DAO and the metadata. And then we are also performing randomization, investigation, product management, regulatory approvals, budgeting, and liaison with the trial site, including the Pakistan and outside of the Pakistan. As a bioequivalence at the Biology Study Center, we are performing the following activities, including the bioavailability study, bioequivalence study, biosimilarity study, and also the alternative of bioequivalence study, which is the comparative decision profile. These are the following objectives of IBBPS. As a PAPS center, the primary objective is to facilitate the pharmaceutical industry in exporting their medicinal product, because we all know that the importance of the bioequivalence report. The bioequivalence report basically support the pharmaceutical industry in exporting outside the Pakistan, especially in those countries that have a very stringent regulation, including the MHRA and SRAs countries. As a CRO, the objective is to facilitate the sponsor in the conduct of the phase 1 to phase 4 trial in liaison with an approved clinical trial sites. As a site management office, the primary objective is to execute the clinical trials in DHS approved trial site. As Dr. Izhar mentioned already, we have 2 approved trial sites, that is Dow University Hospital and Sin Infectious Disease Hospital. These are the certification of IBBPS. As you see in documentary, the basic 3 domains of IBBPS, CROs, clinical trial site, and the bioequivalence, they all are approved from Drug Regulatory Authority of the Pakistan. Second is the ISO 17025 accredited laboratory. The biological laboratory of IBBPS is certified from Pakistan National Accreditation Council on ISO standard. And the last is LGC standard. The biological laboratory and the analysts, they are participating in annual proficiency testing program offered by LGC UK, just to check the competency of the analyst and also to check the reliability of the test result. In all of these certification, we are also enlisted in ICGCP.net CRO list country, just to catch the business outside from the Pakistan. These are the following facilities available at JAO that will help us for the execution and clinical trials and also for the bioequivalence study. They include tertiary care hospital facility. Actually, this hospital has more than 1,000 bed capacity, and this hospital is located in close proximity to the trial site and also for the voluntary healthcare facility, and it helps us in management of the adverse event and also for handling of the emergency situation. And then next we have an institution review board for reviewing the clinical research projects. Also, we have a separate voluntary healthcare facility for the management and for the confinement of the healthy volunteers. We have a pool of consultants available at JAO, and we can take them as a PI and the co-PI, principal investigator and the co-PI. The 2 approved trial sites, this is one of the basic advantages that JAO has, because 2 different hospitals, one is Dow University Hospital and other is Sin Infectious Disease Hospital. These both hospitals are approved from Dow, approved from DRAP, and both are the approved trial sites. And because of that, we are usually offer a larger number of samples as compared to the other trial site available in Pakistan. We also have a satellite trial site option available at Dow, from where we are referring the patient. And the last is we have DAO is the first organization existing in Pakistan that has approved for phase 1 to phase 4 trial. As Dr. Talat Roome explained you about the animal house, and this facility is basically used for the clinical studies purpose. Dow Diagnostic and Reference Research Laboratory. This laboratory basically facilitating us in screening of the volunteer sample and help us in inclusion exclusion criteria. We have a separate access control volunteer confinement area for the especially for the confinement of the healthy volunteers, and we have a separate male and female wards. Okay, the biological lab, actually we have established a separate biological lab in IBBPS for performing the several different functions. The basic function we are performing here, the analysis of the pharmacokinetic sample for phase 1 clinical trial. We are performing here a bioequivalence and the bioreactivity studies. Also, we are determining the different drug concentration level in different biological matters, including the serum, plasma, urine, and so on. And then we also offering impurities, drug impurities in different dosage form, including the NDMA and the EMA impurities. Because this lab is basically equipped with all type of the highly sensitive equipment, including the HPLCs and the LC-MS technique. I think most of them we are all know about the sensitivity of the LC-MS because it's a triple quadruple machine, and this is very highly sensitive equipment that can easily detect in a very minute concentration, even in micro picogram and femtogram, in any biological matrix or in any dosage form. The last is pharmacokinetic and statistical calculation. We are basically using different software for the pharmacokinetic purpose. And these are the following 5 parameters we are determining under the pharmacokinetic calculation. That is the Cmax, Tmax, AUC 0 to infinity, and the half-life. And we are using a Dow Vinolin software and the Kinetica software for the pharmacokinetic calculation. The last is the statistical calculation. We are using the SES software, and we have a very well-trained biostatistician in Dow. And he's very technical person, and he's a very he can easily perform all the above biostatistical calculation by using the SES software and by using different design spreadsheets. Thank you so much. This is my end of the presentation.
May I suggest that we please spare some time for the discussion at the end, Dr. Izhar Sahab?
Dr. Izhar, you are mute.
I would like to add one thing. This facility, this bioanalytical facility is going to be approved by the pre-qualified by WHO as well. The process is going on. I think the next speaker is Dr. Maria. Please.
Yeah. Good evening to all. Let me just. Yeah. I hope I'm audible and visible to everyone listening to us. My name is Maria Eugenia Rizvi. I'm a pharmacist by profession. I've done my master's in pharmacy from Philippines. It is an honor for me to work with Dow Institute of Life Sciences. I have been overseeing and governing the entire institute for the past 3 years. Now, to give you a little profile regarding Dow Institute of Life Sciences, it is also located within the campus of Dow University of Health Sciences, where IBBPS is and DIABAR is. We believe in 4 core values, and that is respect, quality, agility, and integrity. We respect individuals and our business associates. We ensure the best quality in everything we do. We show agility in all aspects of our business, and we strictly maintain high integrity.
Maria, your slides are not visible. Your slides are not changing.
Is it now okay?
To me, no. Okay.
I'm already sharing it.
Okay.
Yeah. My next slide is, yeah, we believe that, you know, in Pakistan, Pakistan should develop and establish self-sustenance, especially in the manufacturing of sera, biologicals, and vaccines. If we are able to succeed in achieving in bulk manufacturing of sera, vaccines, and biologicals, it will reduce Pakistan's dependence on import of biological and vaccines from India and other countries. It will help toward the self-sustaining production of biological and vaccine preparations. It would reduce the cost of procurement and expand the net of biological and vaccines in Pakistan. It will also help provide holistic solution for the EPI and beyond. What is Dow Institute of Life Sciences? Dow Institute of Life Sciences is a biopharmaceutical facility with a dedicated technical team comprising of pharmacists and chemists that guarantees the quality, purity, and consistency of its products manufactured in the site that adheres with good laboratory practices and current good manufacturing practices of Drugs Act 2012 and WHO standards. Dow Institute of Life Sciences, having the assistance and the expertise of the DOHS R&D team, as per my colleagues, Dr. Talat Roome and Dr. Sadia and Dr. Mushtaq, we can develop and introduce unique disease-specific biological drugs and vaccines according to the need of the consumers. Quality systems at DILS starts with the research and development lab, for which is responsible for developing and establishing the stability batches. In our R&D lab, we have a dedicated centrifuge. We have a lyophilizer, lab scale lyophilizer, and a biological safety cabinet, which is BSL-2. R&D lab has its own lab scale lyophilizer, as you can see in the picture. Immunochemical and wet chemistry lab. We have ELISA, gel electrophoresis, Western blotting, HPLC instruments.
Maria, your slides are not flipping, you know.
With me, it's moving, so I don't know really how am I gonna do this?
Okay. Okay,
Yeah.
just.
Dr. Talat, can you
2 minutes.
Maria, can you bring it on the slideshow? So might be then it start moving.
Where is your slideshow? Yeah.
Yeah.
Now?
Yeah, okay.
Yes,
Okay.
Yes,
Yes.
now we can see
Yes.
it.
Okay, so I'm very sorry for all this. So these are the ELISA and SDS tests instruments that we do have in our QC lab. We do have a microbiological lab because you see it's very important that any finished formulation which is being manufactured at our premises have to undergo BT tests and sterility tests. So we do have a biological safety cabinet, which is BSL-2, oven and incubators. Coming to a production plant, it starts with a water treatment plant for which we have a reverse osmosis with EDI plant, water distillation plant with a capacity of 2,000 liters. In our production section, Dow Institute of Life Sciences is the only institute, is the only facility that has the centrifuge RIRA equipment with a capacity of 11.2 liters. This RIRA is important for purification and fractionation process of biologicals. We have an aseptic solution preparation area, which has a capacity of 250 liters. We have a compact vial washing and filling line, which can fill 8,000 vials per hour with a volume of 2 ml to 30 ml. We have a lyophilizer from Teflon, which has a 3 cubic meter capacity. We have an autoclave for a small volume parenteral washing and depyrogenation. We have a separate filling line for sera and for vaccines. This is a capsule sealing machine for which it can seal 60 to 120 vials per minute. We have a dedicated labeling machine which can label 60 to 150 vials per minute. So the future plans of Dow Institute of Life Sciences is to be able to import ready-to-fill COVID-19 vaccine to locally manufacture anti-snake venom, anti-sera, for which my colleague Dr. Talat Roome is doing the basic manufacturing. So in our institute, we can do all the commercialization, bulk production for any research which is being done by the DIABAR and Dr. Mushtaq. To locally manufacture human serum, albumin, IVIG, for which Dr. Roome is working on. Technology transfer, we would like to have within 2 years for anti-snake venom sera, tetanus sera, anti-rabies sera, and that this facility should be able to export vaccine and blood products to other countries. And for Dow Institute of Life Sciences to be the first biopharmaceutical facility that is PICS and WHO GMP accredited in Pakistan. We always say that excellent companies don't believe in excellence, one in constant improvement and constant change. So thank you so much. That's all from my side.
Thank you, Dr. Maria. Now this is the time for Professor Mushtaq, please.
Thank you, Dr. Izhar, for having me over here. And I'll try to finish my presentation within a given time frame. So hopefully, is my presentation visible to all?
Yes.
Yes, it is
Let's
visible.
see.
Excellent. So thank you again, Dr. Izhar and the APPNA Merit. My name is Mushtaq Hussain. I'm by training, or as my undergraduate training is in the microbiology, then my PhD is in the genetics, genomics, and system medicine. And all my followed up experiences are also in the genetics, genomics, and system biology. At this moment, I am a professor at Dow College of Biotechnology and holding the position of the principal as well. And I am also a visiting professor at Loyola University, Chicago. So during the last 2.5 years of the COVID-19 pandemic, we at the Dow College of Biotechnology were engaged in the research related to the SARS-CoV-2 that leads to the publication of about 6 papers with a total impact factor of more than 60, and the total cross citation is crossing now more than 400, making some of those papers are the most cited paper, internationally published, most cited papers that is coming from the Pakistan. At this moment, because of the time constraint, I'll be focusing my work, my presentation on one particular research which we have conducted, that is about the about the design and the immunoinformatic analysis of the mRNA vaccine. So what we first actually explore is that the most immunogenic part of the SARS-CoV-2 is actually its spike protein. We all know that. All of the vaccines which are commercially available are targeting that spike protein. And within that spike protein, there are 2 specific regions that are actually being most immunogenic. And antibodies which are being produced in our bodies are actually targeting these 2 important immunogenic regions. One which is called as N-terminal domain, which looks like this. And the other one is called as RBD domain, which looks like this. And this is the entity of the Wuhan strain, the first SARS-CoV-2 strain, and this is the RBD of the Wuhan strain, that is the first, again, SARS-CoV-2 strain. So these are the 2 target region for developing any kind of the vaccine or whatever the vaccines which are being present at this moment of time. Let it be the Pfizer, Moderna, Sinovac, or CanSino, etc. Now, the brown region, which you are seeing over here, is actually the actual immunogenic part. So now, if you compare the VOCs or VOI of SARS-CoV-2, that is alpha, beta, gamma, delta, Omicron, Omicron XE, delta, or mu variant, you will find there are subtle variations in those immunogenic regions of the SARS-CoV-2 virus, different variants of the SARS-CoV-2. So now the Moderna has actually developed a vaccine against the Omicron or the Delta variants. So as the virus is evolving, so what we thought that there would be new construct of the mRNA vaccine. So in my group, we thought that is it possible that if we analyze those variations in the immunogenic region, can we design a hypothetical entity domain or the entity region, the immunogenic region of the spike protein, and the RBD region? So we developed that hypothetical NTD and hypothetical RBD. So these are the 2 basic regions against which our immune system is developing the antibodies against SARS-CoV-2. Once we have developed it, we map the immunogenic regions on those hypothetical NTD and hypothetical RBD. And what we have found that it is actually covering all the epitopes, if you like, or the immunogenic regions of those NTD and RBD. So then we combine this thing with a linker peptide, which is coming from the APPNA1 protein, that is coming from the Epstein-Barr virus, one of the Epstein-Barr virus gene. And we link them together with a signal peptide. The protein seems absolutely fine as far as the structural thermodynamics is concerned. And once we have done that, we predicted its immunogenicity. And what we have found that we dub this particular protein as DAO21. Now, if you can see, just to put it in a very simple terms, this yellow line represent the prediction of the production of the antibodies out of the 1000 molecules of this particular protein. So this particular, the DAO21, the hypothetical construct which we have constructed, is actually producing or predicted to be produced about 12,000 antibodies molecule per 1000 molecule of the antigen. However, the Pfizer vaccine, which is actually being named as BNT162b2, is producing with the 1000 molecule of the Pfizer vaccine, they are producing roughly around between 4,500 to 5,000 molecule per 1000 molecule of the antigen. So it is performing a bit better, much better rather, than the Pfizer vaccine. Then we remove all the microRNA binding sites. So when we have developed those constructs and when we inject in the human beings, our own microRNA is not going to suppress its expression. So we remove all the expression site and we converted this particular, after removing all the microRNA binding sites, we predicted the structure of this particular particular mRNA vaccine. Now, in a layman's term, if you like, there are blue, green, red lines over here. So more those blue, green, and red lines are superimposed over each other, it means more stable your RNA is. So previously you might have heard of it. There is quite much of the issue regarding a cold chain transfer of the mRNA vaccine. So this is actually the RNA stability, if you like, of the Pfizer vaccine. So there are some areas where the blue, green, red lines are superimposable, but there are some regions where they are not being superimposed. And this is the mRNA stability of the DAO21. So it's relatively not only covering more variants of the SARS-CoV-2, but also more stable than the SARS-CoV, it's covering more variants of the SARS-CoV-2 compared to the Pfizer vaccine, that particular construct. And it is relatively more stable as well. So these are the number of publications that has already been coming out of the work which we have conducted. And we have already filed for the patent for this mRNA construct. And this is the team of very fine undergrad and postgraduate students I'm working with. Like, say, for example, this is Anusha Manullah. We call it our Tigress. She worked about straight 16 hours in a lab. Same with the Aisha Aslam, Sanya, Shabana, Ifat, Basma, Aisha, Fozia, Nasser, Dr. Muniba, which is actually very much involved in the treatment of the SARS-CoV-2 patient, Dr. Humaira, that's the vice principal of the Dow College of Biotechnology, and my dear Dr. Nusrat is a collaborator of the Department of Microbiology from the Karachi University. Thank you very much.
Thank you very much indeed. Are we done with the presentations, Dr. Saab?
Yeah, all the presentations have been done.
Wonderful.
So now is a Q&A.
Yes. Well, first of all, thank you very much indeed for really, really very concentrated, high quality work with a lot of hope for the future, etc. I do have a very quick few questions and some comments. Let me just make the comment first. First of all, I think in Pakistan and in our parts of the world, talking about money and research money and work is a little bit of a taboo. But I think it's very important that the researchers are appropriately rewarded because they do a lot of work which is non-revenue producing, and therefore, because of lack of appropriate reward and compensation, the people do research mostly for love and and they cannot, it's very difficult for them to make a career out of it. And in that regard, I just wanted to find out as to what is the level of funding and whether the institutions that you have are self-sustaining at this time in terms of the revenues, and whether it is going to be a profitable venture for the individuals as well as the institution and the country.
Dr. Saab, it is very difficult to get the funding. And for funding which Dr. Talat got, we have worked about 2 years, and there was so much work and so much problems. Initially, it was rejected earlier, and then with the help of friends, they understand what is the requirement from the funding. The funding is a big problem. Funding is a big problem not only for the researchers that they are not getting anything, but also for getting equipment, for reagents. All these things are problem. So because of the dollar rupee exchange rate, there is a problem has multiplied several times.
Has there been any funding from abroad from Dr. Saab?
I think funding from abroad is also a problem. At this point of time, Pakistan, I should not say anything. You can understand that what is the situation
Right.
of Pakistan. Yeah.
And also, are there any earnings from abroad for the work being done? Or
Dr.
expected?
Mushtaq, if you, as far as Dr. Talat has some funding from the local industry, but not from the foreign industry, I think. And Dr. Mushtaq, if you can throw a light on it, whether you are getting some funding from abroad.
Yes, we are actually the collaboration with the Loyola University Chicago and University of Pennsylvania. So what we have got is some amount of the money in order to develop a human thrombosis model in the fruit fly for the screening of the therapeutic interventions. So we are not only getting the monetary some amount in terms of currency, if you like, as well as some of the consumables are straightly coming from the Loyola University. Similarly, there is a part which is related to the biomarker identification of the vaccinated population of the SARS-CoV-2. So all of the consumables are straightly coming from the Loyola University. So that kind of a support is there.
I think I will be very strongly recommend that I think the group should work with Dr. Izhar to find ways to generate revenue as a result of their products and their functions, because Dr. Izhar is probably the most well-connected person that I know in human sciences. So that you can become self-sufficient and your earnings can support the research as well as the working workers. So I'm now going to open the for discussion and questions by others. They can ask written questions. I think Dr. Izhar has been monitoring those, sir.
Sir, I would like to say one thing. Want to add. For example, Dr. Talat Roome is waiting for Vero cell line, and initially it was rejected by the State Bank of Pakistan, and because of that, the anti-rabies vaccine project may delay because that particular thing is not available. So I just wanted to add this thing before we go for the quick Q&A.
Yes.
Thank you very much, Dr. Hussain.
Thank you, sir. So Izhar Saab, if you would, sir, bring the questions up or comments, sir.
Thank you very much, Dr. Izhar and the team. We are really impressed with the work, and particularly Dr. Mushtaq. So what a great research is going on. So we are very excited. So I'm happy also to introduce to you the Professor Dr. Akhtar Hamid, who is from University of Toronto, and he's a president Canada Pakistan Research and Development Council. So he's kindly joined as well. He's an old Dow graduate. So Dr. Akhtar, can you unmute yourself and please share your views about this vaccine and the work which is being undertaken in the research and development at Dow University of Health Sciences.
It was very eye-opening for me that being an old Dow graduate and being a president, I was trying to connect and get some funded research in Pakistan, especially Dow, and one of the heparin trial, and we had quite a bit of difficulty to get through it and through the trap as well at the time. I'm talking about 2 years ago, just the beginning of the COVID. But now I see the presentation and the labs and all the documentary. It's really opening eyes for us. There, no, we work together. So yes, I would be happy to connect again to Dr. Uzair, and perhaps me and Dr. Uzair will be able to bring some. We also brought the Manitoba University trial to Pakistan, especially Indus, and even we try to get through other hospitals. So yes, we should do something because we have very quality. What I see, there's a very quality control and cost-effective trial centers. And obviously, Canada is looking for cost effectiveness, but without compromising the quality of the trial. So I'll be happy to connect. Thank you.
Thank you very much, Dr. Akhtar Hamid. And Dr. Sarwat, I don't see much of the question. So generally people, they are networking. So we have requested to share their LinkedIn profile because there are people with similar interest and they can contact with each other. Coming back to the main question of Dr. Izhar about the funding, when I visited, I promised that we will help to find the cell line. So in the sideline, I think we will discuss and we will see how can we help them to get connected, because if you know that the CanSino bio-COVID vaccine use the lineage from Canada Medical Research Council. And I can request Dr. Hamid Akhtar and we can really see what can we do on this behalf. So that's very, very impressive. And I think we are just dot on time. So I will request Dr. Sadia Hussain really to formally thank everyone. And
well, thank you very much indeed. This was really an eye-opening and heart-opening presentation, very high quality, very well articulated, and extremely well presented. And I can see Dr. Talat is a Fulbright scholar. I was very envious of her. She did, and all the presentation are really, really wonderful. And I think we have heard Dr. Izhar before also at this forum, at the presentation, and now his team is bringing up the whole scientific and research of in Pakistan, and highly grateful to everybody. I was kind of charmed to see that you have a snake charmer on your faculty, and that the venom can be used for cosmetics also, which is which is wonderful to know. And I think only it can be done for our lady partners, the venom and the cosmetics. With that, thank you very much indeed, sir. I'm most grateful, most humbled, and extremely hopeful for the future of Pakistan with researchers and motivated people like yourself. Thank you.
Thank you. Thank you. Thank you very much.
Allah Hafiz.
Allah Hafiz.